weigh in how much you gained or lost. Right, I believe you can gain. Alright. So before we start, let's have a short prayer. So let's just bow our heads. Father, we thank you for this time. We want to honor you, Lord, as we have our lessons about biochemistry, how you made us and made us to fearfully and wonderfully made us. That, Lord, you guide us and you keep us also healthy as we learn more about your creation, about the human body and everything else. All for your glory, Lord, in Jesus' name we pray. Amen. Just trying to figure out if I could do it wirelessly, but I guess still trying to figure it out. Just before, and when I was teaching downstairs, we could do it in a screen mirror. So I'd love to walk around. So a bit of old school, but yeah, we'll get there. Okay, so this is the start of our topic for the whole year. You have those topics. As for the syllabi, actually, the topics to guide you. You have the chapters, right? You got the chapters as well. The chapters are deleted, so you can use those objectives in the chapters as your guide and what to focus on. But if you listen to the lecture or the upcoming lectures for the whole school year, you will be able to get some snippets of what will come up in the exams, what are the topics you need to study on. Okay? Because I know that biochemistry is a very technical subject. You might expect a lot of memorization. So who can say that they're confident that they could understand biochemistry in one life? So I doubt it's not. So that's why it's like a series of readings, rereadings, and other sets of readings. So according to my teacher before, when he introduced the subject to us, he said that he's need to read 5 times at least. But throughout my student years, I only have read once or twice. And that's it. So, is it possible? I guess it is. If you just be very diligent in understanding. But there's one thing that I've learned. Study smart. So that's why note-taking skills is very important, very essential. If you've got your gadgets with you, use that wisely. Okay? So I should hope that as we are going to be discussing, you won't going to be watching and playing these games. Because it's your loss, not ours. Okay? So that's basically the consequence. But here in when you study smart, you need to take down notes. This is what I would always say to my students way back a few years since I think I started teaching here in 2018. So I would always tell them, bank on your long-term memory, not on the short-term. It's the long-term that remembers. It's the long-term that gives you the understanding of any subject for that matter. Okay? So how would you bank on your long-term? Do not cram. Take down notes, note taking. Definitely keep things in mind. Not just highlighting blindly. Okay? So highlight only the necessary information. So that's why lectures are here to supplement that, and the rest of your readings. Okay? So how can you read such a thick book? It's a very thick book, right? Mm-hmm. But can you finish it? Yes, you can. You can finish it. You just, scroll it down. maybe 1 hour to finish all. So I'm confident to say to my class, they would always ask, or somebody would always ask, have you finished reading this chapter? I would always brag, I finished the book. I finished reading the book. Okay? But, yes, just focus on the things that is highlighted as we go along through the subject. Okay? Biochem is difficult, yes, but you're going to learn something out of it. Okay? So, I'm also a firm believer of short lectures with breaks because attention span will only just be 15 minutes. Okay, so we're trying to keep our lecture short as possible. If it's fast-paced, you just have to tell me, Doc, can you repeat that principle? Can you repeat that again? You just raise your hand. Okay? Just you don't distract your classmates. Okay? If you're doing something else. Alright? But don't let us catch you. Some doctors, some of your professors are strict. Okay? But in this case, let's just have fun. Okay? Alright. So introduction first. These are the easy parts. So this is just gonna be a run through of what happened over the past centuries in biochemistry. Okay, so this is gonna be our outline about history, the important contributors, and then how to link medicine and biochemistry, what are also the recent advances, and also the basic concepts in general of organic chemistry. So this is just a refresh of, or a review of your prima days. So first, our objectives. Is it clear? Not so. The back right? Yeah, our projectors are as old as you. Guys, I think we need to replace the projectors. We will try to log in on that. But I'll be sharing the links to this later. Okay? So, but write your notes. Okay, so first, we're gonna talk about biochemistry in the terms. It's the study of the chemical composition of the living matter and the biochemical processes that underlie life activities through growth and maintenance of that growth. So even before studies about biochemistry started, even before the 19th century, you can see. But of course, at the advent, if you're a history buff, you would know that during the 1600s, you have Robert Hooke. Okay? So he discovered the cell. And then you have also another Robert Boyle, who discovered the element hydrogen. And the other elements by certain notable people. I think for biology majors, you know these people. You have an idea. Or even through high school or a bit of your undergrad studies. So you have also these people like Louis Nicolas Coppelin and Pierre John Robitaille. They discovered asparagine. And Friedrich Wöhler discovered or invented the urea formation in vitro. So you also know that even during the 19th century, there's already the description of the nucleic acids, the chromatin. In fact, Walter Fleming is the father of cytogenetics. Okay? And the third enzyme that was invented by a certain Wilhelm Kuhn. I don't know how to pronounce their name, so let's just treat it as it is. Okay? And biochemistry was then formally defined or termed by Mr. Karl Berg. Then we also have the study about enzymes. So they invented this curve. Okay, so by Unor, Kellis, and Mon, Unor and this is Unor and this one. Then the ATP is described or best the one who described this ATP is Mr. Carl Bowman. Sure, they're Americans, some are Germans, English. Okay. The TCA cycle, that's why it's known as the Krebs cycle, because it was discovered by Hans Krebs in the year 1937. Are they still alive? Could be. During maybe their 90s, or I think they're dead. So, but during the 20th century, you also have your modern scientists as well. So we have also the Gustav and then automated pathway, which is simply the diagnosis pathway. And then the couple goals, 4 couples here by the way. Raise your heart down. So, story cycle, okay, so this is a linear change of the liver and muscle. So it's discovered by a couple, Ferdinand and Gertrude Gordon. I think there's also the one that you discovered, Marie Curie, her husband is also a scientist, they also work together. So that could also happen. Bubbles. Don't worry, just make up a bitter. Okay, next would be the Chargaff's rule. So what's the Chargaff's rule? A is equal to T and G is equal to C. Okay. So that's the Chargaff's rule. And then either Ulrey describe the origin of life. Which for me as a Christian, I don't think that they kind of did it in a scientific manner. But double helix is described by James Watson and Francis Crick. So I'm just like getting to that and what happened over the years. Then the genetic code by Marshall Rosenberg and Henry Maffei. These are American and German. I think Henry is German. So then in the 1970s, we have the central dogma of molecular biology. So what's the central dogma? So DNA is what? It becomes it's transcribed, become an RNA is translated to proteins, but then the surface one of the building blocks, right? One of the things that are needed. And then that's why even for the present times, you have genomics, you have the human genome. Even maybe for I think some of you are the babies, the human genomes are really completed. But right now we're into the proteomics, glycomics. So it's like a very detailed specialization on each molecule or molecular studies. So it's really complicated now. But yet, in medicine, we need biochemistry. We need it as part of our basic understanding of this subject. It's part of how to be able to know diseases, know how to treat these diseases, and know what to give. Okay? So, 4 pharmacists here, by the way. Can you raise your hand? One only shy. Raise it high, I cannot see. Okay, so we've got a lot of pharmacists. That's nice. So you know very much that even if it's part of your major, you have already been bombarded with chemistry. Right? We're even taught how to make, make make medicine, make soaps. Even at the start, I remember during that time, college times, we were usually paired up with, during like first year, second year, paired up with other colleges. We make soaps and stuff, vinegar. So it was easy back then. You just have the materials in there. But pharmacists, they're trained to do so. But mind you, chemistry is really important in medicine. Okay? Research, making vaccines, very important. So that's why it's best described, or biochemistry is best defined as this. It's the study of chemical constituents of living cells and the reactions and processes they undergo. So it has many branches, and these are just examples, by the way. So you've got cell biology, molecular biology, molecular genetics, and so on and so forth. And there are a lot of branches as well. So you've got a focus on proteins, on carbohydrates, on lipids, on immunology, okay, it's also part. But then the main purpose of biochemistry is when we would know how to describe and explain, even in molecular terms, all chemical processes of living cells. And you might ask, Doctor, how can what's the point of studying biochemistry if I'm treating patients? Surely you cannot explain it to them. This is what happened with your carbon, with your protein, with your lipids, right? Will your patients understand those things? Unless if you're talking to a patient who is also in the same or similar field as yours, but it's hard, right? But if we don't understand these things, how can we convey or even share in simple terms? And then it's just the same with quack doctors who will say, it's so happened that there is bad air. Right? So that's why we need to focus on our basic subjects. Okay. So not just biochemistry, histology, physiology, anatomy, these are all shortcuts. So don't do any favoritism with your subjects, because if you do so, you might lose one and lose everything. Okay? That's why it's integrated. Our topics are integrated, so have a balance. Okay? So in your book, in Harper's, I think this is shown, biochemistry and medicine. For example, we will talk about our the basic molecules, proteins, lipids, the carbohydrates, nucleic acids. In correlation to that, if there are defects in the nucleic acids, we get genetic diseases. Proteins, example here is sickle cell anemia, but not just that, a lot of diseases occur due to problems in protein folding, there is acid protein degradation, there are abnormalities in the formation of certain protein, enzymes, enzyme deficiency, a lot, not just sickle cell anemia, it's a lot down the road. So that's why, even in lipids, we would learn that if we eat too much fat, we've got too much fat in our blood, okay? It would create, or it would form plaques in our major vessels, and if these vessels are blocked, it would cause end organ failure, especially the heart, kidneys, and other major organs. So because of this, atherosclerosis. Carbohydrates, of course, the famous disease, one of the common diseases that we have, the non-communicable diseases, is diabetes mellitus. So not just that, a lot of diseases that has also is also associated with carbohydrates. Even cancer is associated with that. Okay, that's why diabetic patients also have a high risk of developing cancer over time. Okay. So what then what are the basic concepts for me? Okay, sorry for that. So now are you still okay? Okay, let's dig in with some basics in organic chemistry and see there. And look for our pharmacists because they're the genius of this part. Oh, no. Somebody raised it. Okay, so what is anything that occupies space and has mass? Okay, substances that can be broken down any further. The smallest unit of an element. Atoms that are chemically bonded to other atoms. Molecules composed of two or more different kinds of elements. Okay. So basically, well, we started our first about organic chemistry, first year, right? Remember that? First semester in your first 2 years in college, you have inorganic chemistry, correct? Remember those subjects? I still remember those subjects. That's a subject, not a subject. Then we have organic chemistry. Then we have also the different branches. Then biochemistry. So there. Then in organic chemistry, of course, everything else in the periodic table. So, but of course, we're going to encounter those inorganic compounds in the body because they're also present. But at the same time, our bodies also has organic compounds. So, some differences that you would know. So both could form bonds, okay? Although for organic compounds, they cannot form quadruple bonds. And then we have for inorganic compounds, variable coordination numbers. For organic compounds, it's constant. Then the geometry is easy to get or to know the patterns because it's fixed. But in organic compounds, the patterns are variable. Sometimes it's large. So, another, atoms. So, what are the three basic parts of an atom? If it's negative charge, you call that your? If it's positive, then if it's neutral or zero charge. So it's the thing that makes each element unique, wherein you know the number of protons. Because the number of neutrons and electrons will vary. So you will know that this is the element based on their number. And if you're going to look at the picture here, so your protons and neutrons are always in the center of the atom. Then the rest are, the electrons are found busy around the nucleus. Okay? In the areas what we call as orbital. So that's why, if you remember, there is that picture. I'm going to show you the picture later. So then how about bonds? So just review. So you've got ionic, covalent, hydrogen bonds. Okay. So when you say ionic, so what happens? There is transfer of electrons between two atoms. So best example there is that your sodium, your So the chloride, right? So transfer of electrons between two atoms. Covalent is when there is a sharing of a pair of electrons between two atoms. And hydrogen bonds when there is you need hydrogen to attract two atoms. Okay? So that's why you've got this. And note also the number of the bonds. It's a single bond, double bond, or triple bond. Which one is stronger on the two? Which one is stronger? If you're gonna learn. Single, double, triple. Which one is stronger? Why? Because you need to break three times, right? So it's just the same principle with a rope, no? A strand of thread is weak. But if you make a lump of a strand of thread, so it becomes a rope in a way, it's stronger. Same principle with that. Okay. So take note, your carbon can make up to even triple bonds. But whereas hydrogen can only use a single bond. That's why hydrogen, when you say when you read in a process that is this molecule is or this compound has hydrogen bonds, you will know that it's weak because hydrogen can only make single bonds. Oxygen can make single or double bonds. Nitrogen, same with carbon, can make single, double, or triple bonds. Sulfur can make single or double bonds. So actually, these are the common elements we encounter as we go along. So what is ionic bonding? So again, it's going to be found in inorganic compounds. So this is what I showed earlier. There. Right? So your ionic bonding will occur when metallic elements bond with non-metallic elements. In this case, it's sodium and chloride. So metal atoms have only a weak hold on their outer shell electrons, whereas the non-metallic ions can have a stronger hold and tend to remove the outer shell of the electrons from any metal ions nearby. Thus, this causes ions to form. So then you have your electrostatic forces. So these electrostatic forces will then pull the positive and negative ions together. Thus, you will form a strong ionic bond. Each of this is surrounded by ions of the opposite charge. Diba? Opposites attract, then the similar would repel. Correct? Is it the same in relationships? Okay. Okay. But it's fun to relate it to real life scenarios. My apologies if you're reminded of a certain situation. So, then each ion is surrounded by ions of the opposite charge, thus building up a three-dimensional structure in which there is what I showed earlier, we call that a lattice formation. Okay. How about covalent bonding? So usually we encounter these bonds, that's why you need to remember that. Covalent bonding, you find them mainly in organic compounds. So it involves the sharing of a pair of electrons between two atoms. And this interatomic linkage will result wherein the binding would arise from electrostatic attraction of their nuclei for the same electrons. And when a covalent bond forms, these bonded atoms will have a lower total energy when that of widely separated atoms. So most of this would include the molecules involved would include your hydrogen, nitrogen, chlorine, water, ammonia. Okay? And these organic compounds are usually the ones that you can see in certain reactions. So there are two main classifications. We've got your non-polar and your polar covalent. covalent. So when we say non-polar, so if the atoms that form a covalent bond are identical. For example, hydrogen, you have your chlorine, your chloride, and then our diatom, nucleus. So that the bonds here must be equal. With the sharing must be equal. Okay? So, and then polar covalent bond, wherein the atoms are linked by this bond is different. So, the bonding of the electrons are shared but no longer equal. Instead, one is more attracted to one atom than the other. That's why there's going to be a shift of electron density toward that atom. Just like in relationships, it makes sense actually. You know, non-polar, there is equal sharing, right? Equal trust, equal sharing of finances. But if the other is the one that is excessive and the other one is not, so there is an imbalance. Can you relate? No? But it makes sense. It's not, I'm just not thinking for any other, with your friends, with your family members, okay? That's why I said in general, in relationship. Right? It makes sense? Yeah. Don't tell me how you're gonna tell us, Mama, we have not covalent bonding. Okay, hydrogen. So hydrogen, if an atom of hydrogen is attracted to two atoms instead of only one, okay? So the bonds in hydrogen will attract molecules to each other, but then they do not practically bond atoms together. So for example here, so the electrons are shared equally by the hydrogen and oxygen atoms. So sometimes they negative charge will form at the oxygen end, and then the positive will form at the hydrogen end. Okay. So here, the DNA. It's a perfect example. So where do you think the hydrogen bond is? Okay, it's in here, right? So it's in the middle. As you can see. That's one example of your hydrogen bond, your DNA. So usually, in the order of strength, okay, hydrogen is weak. It's weaker than the ionic bond or covalent bond, but it's stronger than your van der Waals forces. So, next. So remember this rule, octet rule? Are you familiar with this one? So according to the octet rule, if all elements are most stable when their outermost shell is filled with electrons, okay? And this will be then what we call as energetically favorable for the atoms because it will make them stable. But since not all elements can have enough electrons to fill their outermost shells, that's why atoms will form chemical bonds with the other atoms. Then thus they would obtain the electrons they need to have that stable configuration. So when two or more atoms are with bond each other, their structure is what we call your bond fluid. Okay? So water is the famous one, the famous example. So in here, you would note that some atoms would donate, some atoms would accept, okay, just to fill the outer shells. That's according to your octet rule. Okay. Now we're gonna just have an overview of the different reactions. So further we will talk about this in further details. Okay. So chemical reaction. So when we say chemical reaction, It's when two molecules, compounds, would combine to form a new product. Okay? So metabolites will form new products. So for example, it's synthesis. Okay? So for example here, notebook. So note plus book becomes notebook. That's usually the typical synthesis reaction. So joining of two components that were separated before. Then, for example, bookworm. So some reactions is wherein you would decompose. So I call that a decomposition reaction. So bookworm as one word, then if you break it down, it becomes book and worm. Two different words with different meanings. Unless the word is the book. Sorry, you didn't get the joke. I apologize. Last would be an exchange reaction. So you would tend to exchange this third notebook plus another word. It comes in process that would catalyze, and then you would get that worm is now found in or connected to book. So that's kind of like there's just an exchange of that. So there are also complexes, like for example here, double replacement reactions or double exchange reactions. So ABCD plus EC plus ED. Some things like that. Okay. So that's it for your chemical reactions. Now let's talk about organic chemistry. So before we go for biochemistry, we were introduced about organic chemistry, and the focus here is more of carbon. That's why it's called carbon-based life. So usually, all living things on Earth are formed mostly of carbon compounds, and organic compounds become such when carbon is bonded to hydrogen and other compounds. So you also have your hydrocarbons, the simplest organic compounds, containing only the elements carbon and hydrogen. So you can either have aliphatic hydrocarbons with all the single bonds. So part of that you can have your alkanes, alkynes, also have the cycloalkanes. Those are the aliphatic hydrocarbons. Usually it could be straight chain or branch chain. And especially feature of aliphatic is that they don't have aromatic rings. So aromatic hydrocarbons are the ones with the rings, the benzene rings. Okay? So the longer the chain, the less chemically reactive the compound is. If the compound is less reactive, it has high melting point. So examples here, I hope it's not so clear anymore. But that's like benzene, toluene. Okay, sometimes they end up with that suffix E and P. Okay, so functional groups. You can just read through that or browse the internet for this. Okay. So that's just the first part, okay, of our topic. So let's go for another harder part. But I'll give you 5 minutes to breathe. Okay? Alright, 5 minutes.